Good evening and welcome to tonight's lecture, our first of 2025. I'm George Kraw and I'm very pleased to welcome back Dr. Ari Friedlaender, who spoke to us, I guess, about 3 years ago. Tonight he will be introduced by Dr. Colleen Reichmuth, who is a senior scientist at the National Marine Institute here at UC Santa Cruz, and also a triple slug, having received her bachelor's degree, master's degree, and PhD from Santa Cruz. Dr. Reichmuth, I turn it over to you.
Thank you. I'm going to have to take the hand off. Hi, everybody. This is going to be fun. I just want to start by thanking each of you and all the people tuning in online for being here with us tonight. I know it's a difficult and an emotional week for many of us, and it feels good to come together to champion the worthy causes of science, nature, and our shared environment. So thank you for being here. Tonight's speaker is my longtime friend and colleague, marine mammal ecologist, Dr. Ari Friedlaender. Our lives are interconnected in many ways, and it is my real pleasure to host Ari tonight for the special Kraw Lecture. I know you have had a chance to have a look at Ari's short biography, which touches on some of his incredible accomplishments in research, education, conservation, and resource management. So I just wanted to share a few quick personal observations so you can get to know him before you have to listen to him for the next hour or so. So for me, Ari is a bit like a character in a Jack London novel. He is a rugged guy. He lives a life of great adventure in some of the world's most wild, remote, and very freezing cold places. Ari has crossed the treacherous Drake's Passage nearly 100 times. He's spent almost a quarter of his adult life since he was 20 years old at sea. And I think that kind of qualifies him to be a marine mammal himself. Quarter time, it's pretty good. Ari has worked all over the world, observing, tracking, tagging, some of the largest and rarest and just most wonderful animals on planet Earth. And he balances his time out on the water with a lot of thoughtful analyses and work done on shore and some really interesting and creative conservation collaborations, which he's going to talk to us about tonight. So a few fun facts about Ari. Ari was a professional minor league baseball player. There you go. He has a beautiful family and a lovely wife who happens to be my best friend and my research partner. So that brings us together for sure. Ari loves a good glass of wine. So I see he's already ready to go for tonight. And Ari's also really one of the very best wildlife photographers that I know. And I love to see the natural world through his lens. He's incredible. So hopefully Ari's going to share some of his wonderful images and stories with us tonight, in addition to a lot of good science. And we're going to have some time for questions and discussion after his presentation today. I'm going to moderate some Q&A. So for now, please help me to welcome Dr. Ari Friedlaender. Thank you. I'll put this somewhere.
Well, thank you, Colleen. Thank you, George and Rafe and Nicole and everybody for helping to set this up. These are really quite big honors for us in the faculty at UCSC to be able to come out and talk to folks in the community and folks that have a relationship with the university about the things that we're doing that we think are important, and sharing them with you, and hopefully engaging in some conversation about how our science can be meaningful, the things that you're interested in, and how we can then make sure that that makes its way into the way that we communicate our science. One of the things that I have really enjoyed and am proud of since I've come to the UC Santa Cruz is being able to collaborate with a lot of different people across a lot of disciplines. And one of the most exciting ones for me currently is being able to work with the UC Santa Cruz Institute for Arts and Sciences. And it's really opened my eyes to how we can be better at communicating information and creating information in a way that tells stories to not just scientists, but to people in the public, where we really need to get our messages out to have a broader impact for the science that we're all interested in. So hopefully tonight, the things that I'm going to talk about will intrigue you, will interest you, get you excited about things. Certainly was exciting for me to get to put together this lecture because, as Colleen mentioned, a lot of my work is in the Antarctic. And I'm not going to talk about the Antarctic at all today. And this might be the first time I've ever stood up on my two legs and done that. But it's only happened because California and Monterey Bay and our coastal waters are so amazing. And to be able to call this area home is really special. And there's so much that we can do here. And since I've started a family and really put our roots down here, I'm 100% invested and committed into making sure that the work we do here locally has an impact too, because I want my family to be able to enjoy the things that California has in abundance, and hopefully you all as well. So with that, California does have quite an extensive network of marine sanctuaries, marine protected areas, and specifically marine protected areas that fall under the National Marine Sanctuaries jurisdiction. And these are protected areas that are designated to conserve and manage marine ecosystems while also allowing compatible uses like responsible recreation and tourism. And you can see up and down our coast from the Farallones all the way down through the Channel Islands. These are the outlines of the National Marine Sanctuaries. We're very excited about this brand new one, the Chumash Heritage Marine Sanctuary, which was just approved and increases the footprint of what marine sanctuaries have in our state. And in order to do research or in order to do a lot of things in these sanctuaries, you need permits that help to regulate and oversee the activities within them. And these are jurisdiction of both the state and federal laws. But the sanctuaries are also very limited. And I remember one of the things I learned when I came down here was I got asked a question about what can you and what can you not do in the Monterey Bay National Marine Sanctuary. Turns out the only thing that you can't do is drill for oil. Basically everything else is on the table. So to think about a sanctuary, I think it's really important for us to start to think about what the needs are of the animals and the ecosystems within them, because I would think of that as not just a limitation of a sanctuary, but it's an opportunity to be able to say, this is what we feel like we need to do here. And if we do good science and learn good things, we have the opportunity to place these kind of regulations. So I'm trying to think of this as a glass half full sort of scenario. And from my perspective, I think studying whales gives us that fantastic opportunity, and marine mammals in general. I'm going to talk about a number of marine mammals. But specifically with whales, there's a lot of good reasons to study them. First off, they're a very good model for fundamental principles and testing them and understanding fundamental things about biology, ecology, and physiology. Hopefully, if anyone was here for the talk a few years ago or online, talked a lot about foraging ecology and energetics. The presence of whale also reflects the emergent properties of healthy ocean ecosystems. And what I mean by this is, if we think about a humpback whale like we have here, or a blue whale needs 12 tons of food a day to be able to survive. If you have a population of whales, hundreds of tons of food to survive. In order for the ocean to be able to support those whales, everything has to be working. It's got to be a very well-oiled machine. Coastal upwelling, nutrients, sunlight, everything has to be working really finely tuned to be able to support whales. So the presence of whales, to me, reflects places in the ocean where things are going really well. And then therefore, sort of on the flip side, these whales can be sentinels for climate change. If they tend to change where they're going and what they're doing, or their conditions change, that should be a red flag to us that something else is happening in the ocean that we need to be aware of. And then obviously we care about whales for their heuristic value, for their societal, and hopefully their ecological value. Whales became, or marine mammals in general, became federally protected in 1972, literally through an act of Congress that did not require them to be endangered. The Endangered Species Act, which came around the same time, required a species to basically have one foot in the grave before any protective measures occurred for them. Marine mammals got protection just because they were marine mammals. So we do value them above a lot of all other animals for a number of reasons. I mentioned their ecological value. One of the things that we've been able to glean recently is an understanding of how whales actually help to recycle nutrients and aren't just sort of the end of a food web, but really can help to enhance and stimulate ocean productivity. And we did this through a study we published in Nature a few years ago, where we were able to demonstrate how much a whale eats in a given day, and based on how much it eats, how much it's recycling in nutrients that typically or otherwise are limiting in the ocean environments where they live. So kind of pardon the French, but you crap where you eat is typically not a good thing to do. But for whales, they literally are seeding and recycling the nutrients to stimulate the plant life that is going to support them in the places they are. So in a place like the Antarctic, one of the reasons we think that we've seen a decline in krill abundance in that area is because we killed 2 million whales in the 20th century in commercial whaling, and there just isn't that stimulated, like 5,000 tons of iron getting recycled every year to help that productivity. So they really are good ecosystem engineers. I love to show this picture. Does everyone know who this patron saint is of life? So if I was wise, if I was smart, I would choose a study species or ecosystem or part of the planet where you could get a $2 pad of pen, pad of paper and a pencil and go out and sit and watch animals. And this is a lot of what Colleen does. She's a smart researcher in this way. Go to Año Nuevo, sit there and watch the animals continuously, right? You get a Danish on the way up at the Whale City Bakery. You're home by lunchtime, right? It's like really nice to do. And you can glean a huge amount from these animals. Those of us that have chosen to study animals that do not spend time on land, even if they're marine mammals, but live their lives underwater, require us to develop a lot of technology and a lot of different skill sets. And so one of the things that's really valuable to me about this community is being able to interact and engage with folks from other disciplines. Right? We don't have the resources as marine mammal scientists, both intellectually or economically, to build all of these tools that we need, from satellites to drones to aerial surveys to passive acoustics to tags to biopsy sampling. We really need to collaborate with other partners that can bring in these skills and these technologies and tools. But just to learn about the basic lives of these animals, we really require all of these different tools. So what I'm going to talk about today are some of the projects that we're doing in California to better understand marine mammals, marine mammal health through a number of these different tools and skill sets. So the first thing that I want to talk about is a grant that we have currently that was awarded through the University of California Office of the President under the auspices of climate resilience. And this is a partnership with the Institute for Arts and Sciences, and my role is to lead a marine mammal research unit here to try and understand how marine mammals in California waters are affected by climate change. And this took some kind of creative thinking, but one of the things we wanted to do was try and understand how the anthropogenic inputs into the ocean and coastal waters from industry and from agriculture are manifesting in marine mammals of different types. So we work here with sea otters, we work with California sea lions and humpback whales, which are three of our resident marine mammal populations. And I just wanted to highlight Dr. Jen Casaboon here, who did work with Colleen while she was an undergraduate here, who is finishing a vet DVM PhD, the dual degree thing, which I don't understand why anyone would do both of those together. But she's a phenomenal scientist and someone who's really sort of forwarded our field of toxicology. And some of the things that Jen has been able to produce early on in her career is an understanding of contaminants in marine mammals. And there are what we call these legacy or ongoing contamination by bioaccumulative compounds, such as DDT, related compounds like PCBs. I'm not even going to try these chemical names, so that's why we have good acronyms. DDT, PCBs, BPDEs. These are things that are somewhat commonplace to us, that we know are chemicals that are produced through industry and agriculture that have found their way into marine mammals. And these things can have impacts on endocrine disruption, they can impact reproductive impairment, or they can suppress immune systems. So they can have some real impacts on marine animals. And what we wanted to do was try and understand, based on where an animal lives and what its community is and how it feeds, what is its chemical load and how much of a burden is it getting from human occupation. So our little sea otters live in a very, very small area. These are sea otters that live almost their entire lives in Elkhorn Slough, just inside of Moss Landing there. And we work with the California Department of Fish and Wildlife and some other folks that do captures of these sea otters to do routine health assessments on them. And so we collect biological samples from them. Then we have California sea lions, which are ubiquitous on the docks and on the wharfs around us, that forage routinely in and around Monterey Bay, but stay pretty close to shore. And then humpback whales, which are seasonally resident here in Monterey Bay. I should mention the California sea lions and the sea otters live here year-round. The humpback whales feed for about 5 or 6 months here, and then I'll show you where they migrate to. But we thought that by having these different life histories, the sea otters that feed mostly on benthic organisms, the sea lions that feed on fish, the whales that feed on small fish and zooplankton might have different profiles based on where they're feeding in the ecosystem. This is just a little video showing you how we collect the samples from whales. From the sea lions and otters, it's from captures, but from whales, we use a crossbow with a little flotation device on the end of a dart, and it's got a little cutting tip on the end of it. And when the whale comes up to the surface, we try and get a sample from just below the dorsal fin. So you'll see here, the whale's going to surface. Hits the whale, bounces off. The whale really doesn't respond. It's just like getting, maybe tapped on the shoulder or a little mosquito bite for these animals. We then grab that small tissue sample, and we can do an amazing amount of science just from that. I can't. All the people who are really smart and know how to do this kind of work can figure out a lot about what's going on in these animals. So what we did is we ran this untargeted assessment of all the different chemical compounds that are in these animals. And this is the CSI. You put the thing into the machine, it comes back out with all of the little blips and profiles and peaks of the different chemical compositions. And what we were able to find was that in samples, we collected samples from 5 sea lions, 5 sea otters, and 5 humpback whales, 137 of these halogenated organic compounds, which are effectively the ones that we create as humans, were detected across all 3 species. It's a lot. 137 chemical compounds. 54 of those, 54 different PCBs, very important, very bioaccumulative and nasty compounds. Some of them are banned. 83 of these non-PCB chemicals fall into 19 other general contaminant classes, from industrial compounds, pesticides, and some things that we don't know about that are unknown. There are 18 DDT legacy compounds that we found. Apparently DDT, as it's called a forever chemical, it does break down into different things over time, but you can basically tell exactly that it's DDT or a derivative of that. 18 different DDT derivatives. And then one thing at the bottom here, this DCPA. This was detected in our humpback whale, especially a male humpback whale that's been very well known for a long time, named Batman. And Batman had this DCPA in it that is a chemical that the EPA just issued an emergency cancellation of as this pesticide. First time in 40 years that the EPA basically didn't or bypassed any process or due process, basically said, this chemical's gone. So we're seeing these things in agriculture, we're seeing these things from industry end up in marine mammals. This graph is mostly a visual aid, and what it shows you, the 3 different groups of animals that we work with, the sea lions, the whales, and the sea otters. And each vertical bar is a different chemical compound, and then the darkness or the richness of the color is the concentration that it was found in those animals. And basically, the take home from this is that the whales are the ones that show the vast majority of the different diversity of chemicals in their system. Again, over 120 of them are found are found in the humpback whales. The sea otters have the fewest, but the sea otters have the highest concentration of those chemicals in them. So you get a big spread and a big diversity of chemicals in baleen whales. You get a higher impact or load in the sea otters. There certainly are some differences between life histories. We know that males will accumulate these over time. Females typically will metabolize some of these and give it to their offspring in milk. So we tried to steer clear of females in this case, just because we wanted to see what the breadth and the scope of the chemical compounds that were in these animals. When we look specifically at these DDT-related ones, so on the left here, you see the DDT-related chemicals, and then on the right, the PCBs. The interesting thing here is that if we look at sea otter blood specifically, it contains a higher abundance of those DDT-related compounds, about 10 times higher, or an order of magnitude higher than the other animals. So the things that they are eating, for some reason, are getting a higher dose of that DDT into them. These are sea otters that are not living down in LA, which we'll talk about in a little bit, where we know there are dump sites. These are ones that are living in Elkhorn Slough and still able to receive quite a high load of DDT. So these animals are getting quite high impacts. The next step, once we quantify this, is to try and understand the impacts and what the actual impact is on individual animal health and then on population health. But from this first spread, this first sort of investigation, we can certainly say things like humpback whales accumulate the greatest total number of individual compounds and contaminant classes, that sea otters don't have as diverse a contaminant load, but they have much higher abundances. And for a small animal, that obviously can have a bigger impact. And sea otters have the highest abundance of also unknown compounds. It's very likely that a lot of these are just making their way into the ocean ecosystem through agriculture. And the thing that we can relate back to climate change or climate resilience is that as conditions change on land, agricultural practices are going to change. The pesticides, the chemicals that you need to use, even the crops are going to migrate in terms of what is being farmed in different parts of the world or in different parts along the coast. As those change, these animals are going to receive the impact of them, and they're going to be the ones that sort of tell the story of that in their tissues. So something that we wanted to set at least a baseline for so that we understand how contaminated these animals are as we move forward. In terms of how this then links to our marine sanctuaries, I think there's a couple of ways we can start to think about this. These inputs into these marine sanctuaries are coming from land in various ways, through watersheds, through direct dumping, and probably from a lot of other sources that we just don't understand at this point. And like I just mentioned, as climate change affects our agricultural practices, water usage by people in different areas, the location, the timing of runoff, storms, the magnitude and the severity of storms, and as that changes, that's really going to impact these animals. So getting a baseline, like I mentioned, I think is really important so that we can track these things over time. I mentioned DDT, and one thing that happened relatively recently was some colleagues of mine totally serendipitously contacted me about some work that they were doing in Southern California, and I'll kind of get to why this is important, but off of California, off of LA Long Beach. So this is the basin between, that's Catalina Island on the bottom left there, and that is the Palos Verdes Peninsula right there down in Southern California. In this basin, there was dumping of DDT over many, many years. And this group was interested in characterizing a little bit more about this. So from the 1930s to the 1970s, that San Pedro Basin was the site of documented industrial dumping, okay? People literally went out there with barrels full of DDT, dropped them in the ocean. We typically think out of sight, out of mind. They're going to go to the bottom of the ocean. They're going to get covered by sediment. They're going to be fine. We're never going to notice them. Then in 2020, there was also this renewed concern for bulk and containerized dumping of DDT waste and byproducts that came out in a couple of news articles. And so these folks down at Scripps and UC San Diego, who are colleagues of ours, wanted to go out and understand how and where these sites were and what you could characterize about the environment around them. So they went out and they did this really amazing thing. They did these two different types of surveys. They use AUVs and ROVs, so autonomous underwater vehicles and remote underwater vehicles. They use two different types. The type that's up on the upper left there uses what's called a side scan sonar. So it's using an acoustic sonar to map the seafloor and see it through acoustic signaling. And then they did a lot of surveys, and you can see the survey track lines here just mowing the lawn across this basin. Then they used this ROV, which has a lot more imaging and image processing power on it, to go down and literally visualize the seafloor in the places where they were detecting things from their sonar. So you go out with the sonar and you do a survey, you see where you see piles of things or stuff on the bottom of the ocean, and then you go and you do a little bit more documenting with this. They found a lot of DDT, but the reason they emailed me was because they found 78 whale falls. Whale falls are basically the skeleton of a whale that has fallen to the bottom of the seafloor and has created a massive boon of carbon and protein for an entire benthic community at the bottom of the ocean. These things were first discovered in Monterey Bay and the Davidson Seamount, just sort of southwest of us. And there are communities that thrive on these whale bones. So in the first 1 or 2 years, it's mostly scavengers that are pulling off the meat and pulling off the tissue. Then when you get down to just the skeleton, there are specific worms that have evolved specifically to live on whale skeletons and bore into the bones and get the oils out of the bones. And it creates this habitat and this ecosystem in an area of the ocean that typically is just getting very, very small particles that rain down on it. So you have communities that come together and thrive for decades sometimes on a single whale skeleton. We thought whale skeletons and whale falls were relatively rare. We know whales die all the time, but there'd be one here, one here, one here. I've never heard of 78 whale falls being found in the same area. So big red flag goes up. And I'll talk about why this is a red flag. Probably not DDT, but there's something else that goes on in this area that I think is something that we need to look into. But I wanted to show you guys some of these images because this is just wicked cool stuff. So on the left, what you see here, these are these side scan sonar images of what a whale fall looks like. And so what you're looking at are the skeletons of baleen whales here. So for example, on this one right here, you can see this is the vertebral column right here. These are the flippers. This is the upper part of the jaw, and these are the two lower mandibles. So you're looking straight down. That's the front of the whale. That's the back of the whale. And you see them in different sort of orientations and stuff, but this is really what sticks out on those sonar images. And then they had the wherewithal to go down and to actually send the ROV to look at some of these skeletons. So I'll just show you a couple sort of highlighted pictures. This is a really cool one here. So what we've gone through and done is to measure the length and the width of the skull of the animal here. We can go back into sort of the old zoological records and get an idea about what species of whale this is most likely to be based on the length to width ratio of the total body to the width of the skull, as well as thinking about things like, gee, how many whale species get to be 17.8 meters long? All right, that's about 60 feet. There's basically two. It's either a fin whale or a blue whale. So we can start to do a little detective work once we measure all of these and see what the species composition of the animals that are dying in really high abundances are in this very small part of the world. Here's another look. You can see the skeleton a little bit better. Like you see that very long vertebral column. The animal is facing that direction there, and you can see the mandibles and then the middle of the upper rostrum there. Again, this is a 16.8 meter animal. Nothing except a blue whale or a fin whale there. Now here's the goods. This is one of the ROV videos showing what that whale fall looks like. So you can see this is the substrate here. I don't know exactly how deep this is, but 895 meters. Not close to the surface, right? So we're talking about, more than half a mile down. So now you see the vertebral column here. There's some sediment that has accumulated on these animals, but you can also see a lot of life that is on the back of this. I'll let this play. This is just a couple of minutes long showing how we move up and down on the animal here. And it's a great time to drink some wine. So all that snow, that marine snow that you see right now is all the detritus and things that typically are falling down into this environment. So imagine this sort of little snowfall versus a 60-ton whale landing at your doorstep. It's going to attract a lot of visitors. So these are obviously the vertebrae here. You can see in between the vertebrae that cartilage has gone and has gotten eaten. You can see the evidence of lots of little worms and other things that are living on top of this whale. And we'll get up closer to the skull here. So there you have a good look at the skull of this. This is a big, large baleen whale. Really amazing to see these kinds of things. Excellent. So we see in some ways there are whales that are dying and sort of ending up at the bottom of these areas, and I think that's a really important thing for us to think about. Why they're dying there, but also then how animals are using sanctuaries and how they're moving up and down the coast that puts them in these places. And one of the projects that I did recently was partnering with the World Wildlife Fund and a few other universities where we have colleagues to start thinking about the places where whales are any moment in time as being a critical place to conserve, not just the endpoint of Monterey Bay or Mexico where they breed, but anywhere they're in this corridor of migrating between. We need to be vigilant about protecting those areas too, because those risks are real for those animals. So what we did is we organized as many people in our community as possible to provide as much data on whale tracking as possible. So tags that you put in whales that stay in for a couple or a few months that show the routes of the migration over long periods of time. And this is a really nice showcase sort of around the world where all the tracks of whales exist within our community. And then the idea for us was to think about looking regionally at different parts of the world and trying to understand the threats and risks that whales face. And the most major of these are things like fishing, climate change, and at the bottom here you have shipping. And these are the three main risks that we want to be able to think about and quantify when whales are moving between big locations and when we're not seeing them from day to day. So using large databases that exist like Global Fish Watch, climate change indices about how things are changing. And then there are a lot of databases on marine ship traffic. And you can see down here just how much shipping there is around the world. And those places that are really rich red are really indicative of massive shipping areas. Obviously, the coast of California has an awful lot of shipping, and there's a lot of shipping that goes through our marine sanctuaries. And there are some really awesome things that are getting done right now to try and mitigate the risk that whales face from shipping. The main risk that whales face in our waters is from ship strikes. You have these massive container ships that are moving up and down, north and south through the coast, right through the areas that are really important for whales, and getting an understanding of how often those animals are in the same place at the same time is important. These animals have not evolved to avoid ships. A large container ship that's moving at 20 knots isn't projecting sound forward. You really can't hear that ship coming. Even if you do hear a ship coming, as an air-breathing animal, one of the things you do is come to the surface, to the safe place, which might put you right in harm's way. So the best thing that we can do to try and minimize negative interactions between ships and whales, slow the ships down. If the ships are going to be there and the whales are going to be there, make it so that they just bounce off each other instead of run each other down. And so there's this initiative that's been led by the California Marine Sanctuary Foundation called Protecting Blue Skies and Blue Whales, or Blue Whales and Blue Skies, to try and voluntarily get shipping organizations to come to the table and give these voluntary speed reduction zones a chance. And this has been a really fruitful thing. And the idea is to think about where we know whales are and where the large shipping routes are that come right in contact with them. So one of the main ones that we have right here, this is the basin between Catalina Island and in LA Long Beach here. This is the transition station and the voluntary traffic lanes where most of the shipping traffic of the second largest port in the world occurs. Why are there 78 dead whales on the bottom of the ocean here? Probably because a lot of them are getting hit by ships right in this area and they're not making it anywhere else except down to the bottom of the ocean. This has been a really productive process though, and it took a long time to get done. But what this graph shows is the voluntary engagement with different shipping organizations to voluntarily slow down and take these vessel speed reductions seriously. And there's a sort of an incentive program for companies to do this. You get a little trophy, you get a medal. You literally can say to the next competitor, you know, we do good for the environment. We have a certification from the Blue Whales and Blue Skies Initiative. You guys don't. People care about that as a consumer. This is where you're going to ship your products with, yada, yada, yada. You can set up some competitive marketing there. And this graph just sort of shows the cooperation rate or the buy-in by companies, which I think is really amazing in a short period of time. So this is one of these things that gives us hope that without having to make a major change in an industry and not tell someone to not do their job or not make a living, but work with us a little bit. There's ways that we can minimize risks to animals without having a huge impact on industry. And I think that's important. We wanted to start thinking about humpback whales as sort of our most coastal and our most abundant species that live in these waters and start thinking about where these animals go. And up until a couple of years ago, we didn't have a ton of information other than anecdotes and photographs to match where animals go throughout the season. We know animals spend a lot of time in Monterey Bay. We know they go down to the Channel Islands. We don't know how much time they spend in these different areas or how much time they're spending and what they're doing in each of these national marine sanctuaries. So what these two graphs show, these are two slightly different things. On the left is spring and on the right is fall. And we went out last spring and deployed 6 or 7 satellite tags. I think it was 6 tags in spring and 5 in fall on humpback whales in Monterey Bay. And we said, we want to see where these animals go throughout the summer. Where are the important areas that they use? So humpback whales that we tag in Monterey Bay, kind of in the middle there, each color is a different animal. You see some of them wander up a bit north towards Point Reyes, through the Gulf of the Farallones. Some of them go south all the way down to Point Conception here. So we have a pretty broad area that these animals are using in the summertime. And if you look at those tracks, the more kind of squiggly the track, the more time an animal is spending in a certain place. The more straight the track, the more it's just moving out through an area. But what's really amazing is that the boundaries of these marine sanctuaries pretty well overlaps with a lot of the areas that these animals are utilizing to forage in. So great, we've got some good coverage in this part of the world where we can say, what can we do to these sanctuaries to try and minimize the effects of human industry on those whales? On the right, and I'll show you this in a slightly larger view, the animals are still using these marine sanctuaries, but they're starting to do things like move farther south. And so knowing that the animals just aren't there in the springtime or just aren't there in the summertime, but are using these areas dynamically is something that we can then take to conservation planners and say, hey, when animals are here at this time of year, this is what they're doing and this is the risk that they face. When they're here at another time of year, they're behaving differently and they may have other risks. This was the updated map from yesterday. We have these data are all up online. If you want to go to the, oh, you're starting to read the names, sorry. Grappa really did, yeah. You got all the way. So we tried to do wine names in the first deployment. You can see we got into the heavier alcohols in the fall. It definitely follows our mental state a little bit. But this is a really cool graph to me, and it shows this is sort of the entire range. This is the blue corridor for humpback whales that visit Monterey Bay to feed. They're feeding from Monterey Bay all the way up to Point Reyes, all the way through Southern California, and then you see where those lines start to go straight. That's migrating down here. So this is the Baja Peninsula, and the animals basically hit land here, and then from here all the way down here in mainland Mexico, these are the breeding areas and the calving grounds for this population of humpback whales. So I like to think of these whales as Mexican whales that are breeding and calving down there, and then are feeding up in our area in California. But what's critical is that regardless of where these animals come from and go to, we now have 2 countries that you need to get on the same page in order to protect these animals. If we do all the conservation planning in the world in California, and by the time they cross the border in San Diego, those protections go away and you don't have those same kind of mitigations, the animals still are going to face a huge amount of risk. So the animals don't see boundaries, they don't see political boundaries or governance. We need to make sure that the countries that these whales visit, or the groups or the counties, the states that these animals visit, are able to get on the same page and deal with conservation planning together. So it's a great way to bring together countries and think about conservation. Now I wanted to switch gears a little bit and talk about another type of study we're doing that brings us a little farther south down in the coast. We've talked about threats from contaminants and threats from shipping. One of the other sources of disturbance for marine mammals, especially in California waters, is acoustic disturbance, either from ship noise, but in this case, we're looking specifically at Navy sonar, which is mid-frequency active sonar that's produced by submarines and sub-finding ships that is used largely by the Navy to try and locate submarines. Submarine, hunt for red October. You want to stay silent, just one ping. Remember that line? Great line from the movie, Hunt for Red October. That ping is a really loud sound that goes out into the water. It's going to bounce off of something. That return echo is what you can image and dictates what you learn about what's in the water column. Same thing as echolocation in these animals, that these things like dolphins, they use echolocation to image their environment. It's just a very high frequency sound that they use. So over the past several years, we developed a really interesting approach to trying to understand whether or not dolphins are affected by Navy sonar. We've spent the last 10 years doing this with large baleen whales and doing it on individual animals, and have learned a lot about sonar impacts on them. But what we don't know is how it affects dolphins and very social animals. So there's this very high need for directed studies of these kinds of animals because they are very large numbers, very social groups. And if you're interested in the animals that are impacted by sound the most, it's going to be the most abundant animals. They're very common. That doesn't mean that they are going to be any less important. So Literally, this is called a common dolphin. This is the species of common dolphins, very common. They get exposed to sonar almost all the time. They live in areas where there's Navy operations, right outside the Channel Islands to the south and west, around San Clemente, San Nicolas Island. So these animals are inhabiting a National Marine Sanctuary that is also being insonified by Navy sonar at the same time. And a large proportion of the predicted negative effects of Navy sonar operations are going to impact these little guys right here. Okay, I mentioned the mid-frequency active sonar as well. The challenges that we face with these little guys, so this is one of our suction cup tags that records audio and inertial sensing to give us sort of animal movement underwater. This is on a species called a rhesus dolphin, which is about 9 or 10 feet long. They're a pain in the butt to tag. They don't come close to vessels. And then when you get a tag on them, they oftentimes don't stick on the animals because you see all these scratches and marks on these animals. The animals will literally go and take it off of the other animals if they don't want it to be on there. So convincing these guys to volunteer for our experiments can be really challenging. So for animals that live in these social groups that don't like having tags, the social unit that we're more concerned about is the group rather than an individual. So how do you study a group of animals that's on the go very much, that's very noisy, without being able to put a tag on them? And this was sort of the challenge that we took. And so the objectives of this study were to, first of all, just develop some kind of an integrated method for tracking animal behavior and physiology sort of remotely, and then use those methods to get a baseline assessment of just what these animals are doing, how they're communicating, what their physiology is like, and then conduct these experiments on them to see how those groups respond to mid-frequency active sonar. And so this is research that I mentioned is taking place around Catalina Island, just adjacent to where naval operations are. I should also mention that this presentation I'm moved into right now, my wife Carolyn just gave this at a really cool meeting called The Effects of Sound on Marine Mammals a few months ago in The Hague. And I thought the talk was so well put together, I wanted to just literally take it, import it here, and present it to you guys. So it's a really cool approach that we have here where we use theodolite tracking of behavioral observation. So we have a team that's up on a cliff with a theodolite, which is basically a really high-powered set of binoculars that has reticles and is georeferenced. They're going out and looking from a cliff top where groups of animals are. We then go out in a boat, and I'll show you how we do this, to get a drone up in the air that is going to collect photogrammetry information about every individual, their orientation, their speed, their movement, and it's going to go follow those animals for a period of time. Then we're going to put in these passive listening stations along the way that are going to pick up all the whistles and all the sounds that the animals make, and then as well as it's going to record the sonar playbacks that we do. And then afterwards, we go out and we collect these small little skin and tissue biopsy samples to see if we can measure a physiological response from these animals. We do this looking at cortisol levels. Cortisol, just like in us, is a hormone that doesn't directly tell you how stressed an animal is, but it tells you that there's a physiological response of stress in an animal based on the level of cortisol you have. So the approach was to look at these two different species of common dolphins, short and long beak. They look basically the same. One's got a beak this long, the other's got a beak this long. So telling them apart can be non-trivial, but these were the species that we were interested in. And this is sort of the functional operation. So we have this land-based setup that's on the cliffs in Catalina. They identify a group of dolphins. We then send a boat out after them to try and locate that group. And then the first thing that we do is engage with that group, see how many there are, what's their behavior like. And then we deploy the first thing that goes in the water here is going to be one of these listening stations. So we're starting to record the animals before we do anything else. Then we put a drone up in the air. It flies over the group of animals that we want to target, and it's going to hover over them and move with them. It's georeferenced, so we know precisely where that group of whales or dolphins are. Then we get into a pre-exposure period. We take 10 minutes and say, let's just collect a baseline information on what these animals are doing for 10 minutes. As they move, we put more acoustic recorders in the water around them. Then we either on our boat have a simulated Navy sonar source, which we put in the water, and we play with our mid-frequency active sonar, a set number of pings for a set duration of time. Or we coordinate with the Navy and we ask them to bring one of their helicopters, and they put their sonar, which they literally kind of drop down into the water from a crane and turn it on, and they use this as called a dipping sonar. So they'll just fly around putting the sonar on the water, turning it on for a ping here or there, doing it every so often to try and locate submarines. So we have both our own source, and sometimes we can get the Navy helicopters to do that with us. We do that for 10 minutes, play them this sound, measuring everything, and then we continue to do this for another 10 minutes afterwards to see if there's any latency in the response or how long that response takes. And then after that, we go into the mode of collecting biopsy samples about an hour after for about 2 hours so we can see if there's any change in those stress hormone levels, like I mentioned. And then analytically, we're looking at things like the speed of the animals, the whistle counts, how frequently they're communicating acoustically, and how much the animals are coalescing into large groups versus breaking up into small groups. And we use these Bayesian hidden Markov models. We look at the baseline state that these animals were in and whether they change to what we call an enhanced state. Does their speed change? Do the whistles change? Do the subgroup numbers change? And try and understand this in 5-second blocks throughout this 30-minute window. So instead of doing it continuously, we're looking from one 5-second block to the other because we're collecting data in really, really high frequency. So we don't want to average over too long a period of time. And what we're looking for is there's this probability of moving to an enhanced state from the pre-exposure, which is the baseline, to when we give them sound, and then afterwards. Basically, the things that we want to also measure as other variables are the received level of sound. So how loud was the sound that the animals received when they started to change their behavior? And what was their pre-exposure speed? How fast were they moving ahead of time? Okay, so over the last few years, we've been able to do a good number of these experiments. We did, I think, 22 here. Group sizes range from between 10 animals up to 800 or 900, so sometimes very large groups. The focal distance to the group is about 1 kilometer away. And then you can see this is the max RL, which is the received level in decibels. It's a relative scale of sound. It's a logarithmic scale. So 1 decibel up is not just a continuous linear increase in sound. It's a much greater increase in sound. But we've tried to get these animals to receive the sound level lower than what the Navy would typically give to them. I wanted to show you guys just what it looks like from a drone measurement, what we're doing here. And I want this to be kind of overwhelming because you have a group here of, I don't know how many dolphins, but you get a wonderful undergrad that wants to sit and look at images that are collected 60 times a second and go in there and identify every single individual dolphin in there. But not only every individual, but what's their orientation and what's their spacing from one animal to the next. And then what we do is every frame of the video, you're able to look at one animal in the video in frame 1 and in frame 2, so on and so forth. And you can actually get tracks of individual dolphins. And so what that allows you to do is look at how that group is moving as a whole, what their orientation is like, what the speed is like. And this is sort of showing you, in this case here, the black is a pre-exposure period. The animals are moving in one direction. Then during the exposure, they kind of stay in the same direction, and then they make a big right-hand turn at some point in there. So this is just a visualization of some of the data that we're looking at. And then in the interest of time, the things I wanted to do were just sort of show you what some of the results were for the things that we've learned about these animals. And the first thing that we've learned is that short-beak common dolphins, they travel slower in a pre-exposure state, or when they were slow moving, they're more likely to speed up during an exposure. So you're going slow, you hear a sound, the response there is to speed up. For long-beak common dolphins, the groups were traveling faster in a pre-exposure phase are more likely to slow down. So it's not necessarily always speeding up, it's just a change. Sometimes slowing down, you're reorienting, you're trying to understand where a sound is coming from, or you're speeding up and trying to get away from it. So these animals are showing some difference in how they respond to sonar through their speed. Another thing was the received level. So in long-beak common dolphins versus short-beak common dolphins, and here you can see if you were to just sort of generate a regression line through there, the greater response occurs at greater received levels of sound. We don't see the same thing happening in a very similarly related species, the long-beak common dolphins. So even for dolphins that may appear to be very similar morphologically, live in the same areas, they're responding to sonar in different ways behaviorally, and that's important for us to understand. And lastly, when we look at cortisol, or the stress in these animals, the two that you see on the left there, the 2 bars that you see there, were biopsies that were collected opportunistically. And the middle one is the control group where we collected samples before the exposure experiment. And then on the right, are ones that we collected directly after we exposed them to sonar. And this is a significant difference here. The stress hormone levels go up. That cortisol level goes up slightly, but significantly. Interestingly, though, like you see here, the cortisol here is far lower than animals that we have measured when they're either stranded or bycaught in nets. So this response level could be just a little bit up and down. Folks that live in the city versus living in the coast have a similar difference in stress hormone levels. You can live just fine with this, but it's an elevated level. It can have chronic impacts over a long period of time. But if you want to think about acute stress in these animals, like being caught in a fishing net, these levels are nowhere near that. So it's at a low level, but it's persistent. And if this is an animal that lives in a Navy range and is getting exposed to sonar every single day, and you're living at an elevated stress level for your whole life, that could have impacts sort of down the line. These are what those levels look like. Sorry, I didn't realize I had this slide in here. These are what those levels look like in bycaught animals or stranded animals that end up on the beach. You can see those levels really rise up quite a bit. So to conclude sort of from this study, what we've learned and what we've been able to achieve is successfully integrating a number of very disparate methods that we typically use one at a time in a remote location. We have tons of information now on the baseline physiology and behavior of these species, and the first ever sonar exposures to this group of animals, which means we can now also go to other places and other species and use this as a tractable method. And like I mentioned, we showed that the responses of shortbeak and longbeak common dolphins, even though they're occurring in the same place and they're very related species, are quite different, speeding up and slowing down, the whistling versus non-whistling, the type of subgroups versus aggregating into larger subgroups. Those things change depending on the species that you have, and we can't have a one-size-fits-all kind of approach to understanding the impacts to these animals if we were to do that. So really interesting and fun work that has sort of taken us into a new direction of how animals that live in marine sanctuaries are still affected by things that happen outside of them. All right, so just to sort of summarize the things that we've tried to talk through tonight, California, first off, is an extraordinary place. It's got amazing potential for conservation of marine species and ecosystems, and we're well ahead of a lot of other places in the world, and I'm proud of that. We're racing to understand and quantify the risks and threats that marine mammals face from human activities and climate change, and we need to set as much of a baseline as we can now so that we understand how these things change in the future. Sanctuaries offer us that opportunity to do that. They offer sanctuary literally from harm from these and other animals, but they're not the best place in the world to get away from all of these things, and we need to be aware of that. I think I've shown, and hopefully it's come across, that collaborative work, integrative work, and creative research and communication are critical, not only to collect information, but to share it and promote it more broadly. And that more than ever, now more than ever, literally after last week, more than ever, it's going to be incumbent on people, not governments, to be able to support this kind of work and to support the things that are important to us in literally our backyard. And I hope we all take that very seriously. With that, I appreciate you guys for listening. I thank the Kraws and everybody at UCSC for letting me talk to you, and Colleen for introducing me, and Ryan for driving me. That was big. So great. I'm happy to answer any questions you have. Thank you. I'm going to put this one back up just because it's pretty. Good. Should I sit down here?
Yeah.
Okay.
Let's see if I can sit down
Yeah.
here.
You got it?
Yeah, step on this one. Thank you. Wow, that was so great. That was really amazing. So we have maybe about 10 or 15 minutes for discussion and some question and answer with Ari. This is a chance to kind of dig into, obviously Ari just kind of scratched the surface. I don't know how he selected these particular 3 projects from the range of research that he does, but it was cool to see these vignettes of something kind of in our own backyard happening with these great whales. So does anybody want to start us off with a question? Okay, let's start over here. Yeah, we're going to give you a mic for the folks online so they can hear.
You started off with a very generous shout out to the Institute of Arts and Sciences, and so this question is more from the art side than the science side. But I was blown away by your graphics, particularly the chart that you, or the image that you showed of the worldwide whale routes.
Yeah.
Could you talk a little bit about your working relationships with the graphic designers who pull that stuff together? I mean, even your ones that came out of a scientist's computer were pretty impressive, but the one of the worldwide whale routes just was mind-boggling.
Yeah, I appreciate that. So that was directly the result of working with WWF, an NGO that does a lot of science communication, and it's a lot of back and forth. And the caution that we have to have in there, or the mindset, is that we're not creating a figure for a scientific publication. We're creating an infographic that is trying to represent a theme, not a very precise set of
People like me.
Exactly to everybody. That everybody should be able to understand. So it was just a lot of back and forth working with their graphic design team and knowing that they've done this before and telling them, how do we do this given the data we have? Yeah. And I think that's, for me, that was the best part of that experience, is learning how to build data that can then be translated in a really informative way. So I appreciate that. Thank you.
It seemed like the Batman whale had maybe 80% of the compounds that you're testing for. Was it just a loiterer around the areas that you were testing? Was there just a lot of dumping in that area? How do you explain such a myriad of chemicals for that one whale?
Yeah, all the whales, all of the humpbacks in there had a high number of them. Batman, we also know, is an older whale. It's at least 26 years old. I think its first sighting from Monterey Bay was like 26 years ago. So we know it's had a relatively long history. We don't know the precise age of the other whales that we tagged. So it could be that those are younger animals that just haven't accumulated over time. Very good question.
Ari, I actually had a very similar question. You have these otters, sea lions, and humpbacks that have really different lifespans, and they're using different areas, and they're eating different things. And, ideally you want to try to tease apart what the drivers are of these contaminant loads. How do you get through that? I mean, for some ways, maybe the otters have it worse, but their lifespans are so short, they're not able to accumulate over a longer period of time. So how do you tease those kinds of factors apart in these projects?
I think it's a real challenge. We're logistically challenged. Luckily, like I mentioned, there were these programs already initiated where they're capturing a ton of animals, and sample size is the best way to do it. And those chemical analyses are expensive too. For a non-targeted thing, I think it was about $1,000 to run. If we wanted to target a specific set of chemicals, though, I think it's about $5,000 or $6,000 a sample to run. So we have to be kind of judicious to what we can afford to do. And those non-targeted ones are nice because they just give you a broad sweep, but that's a difficult question. Yeah.
Having worked with a lot of these animals in that area, I think it's really exciting to kind of go up one level and look at population levels of health and status in like a big area, a big important area of the world. That's great. Can we get a mic up here for Arthur?
Thank you. About the dolphins, are they moving to or from the sound? Towards or away from the sound?
Very good question. For the most part, I couldn't say whether or not it's definitely not toward, but it's not always away. The way that the experiment is set up is the animals are moving in this direction. We put the sound source here, anticipating that the dolphins are going to keep going this direction. Oftentimes, they turn just naturally, and so they're already going away from the sound source when the sound comes. Whether they continue on their path, that can be away from the sound source. Very rarely do we ever see them turn towards the sound source, though. But it's logistically kind of tough to keep them in one place for a long period of time.
And I'll just chime in there as a sensory biologist. It's really interesting to think about sound and mammals underwater because the wavelengths of sound are very big and our bodies are acoustically transparent. when sound is coming in from the water through our fluid-filled bodies. It passes kind of right through us. It can be really difficult to tell which direction a sound is coming from, even for a sophisticated animal like a dolphin. So that's a really interesting question. We're going to jump to take one of the online questions, and then we're going to come back over here.
Excellent. Well, in that case, I'll pick the ones that I was the most interested in, because we got a lot of questions about the whale falls. And I think I can kind of fold them into one kind of mega question and ask, first and foremost, I know that this is relatively new, but can you tell the cause of death from the skeletons based on just observation? And then kind of a larger question, this idea of, having ships slow down in the sanctuaries, why not just make it a mandate, make it a law? Like, is really kind of this approach of incentivizing it kind of emotionally really the only option that we have right now, and kind of how do you guys navigate that?
Yeah, great questions. So when a whale gets struck by a ship, typically there is a lot of blunt force trauma. I've done a lot of stranding work over my career, and with large whales, you very often will see subdermal hemorrhaging. Not always will you see broken bones, though, unfortunately. Just the impact alone can do enough damage to the animals in the soft tissue that it doesn't necessarily break the back or break a vertebral. So it's one of these things where you are likely to see broken bones on the animals, but the ones that don't have broken bones don't mean that they weren't also hit by ships. So it wouldn't be a way to quantify the rate of ship strikes. In terms of slowing down, global shipping, I think 90% of our commerce in the world goes through ships. And I know some of the folks who tried to do these vessel speed reduction things are working with the IMO, the International Maritime Organization, to try and think about ways to get industry buy-in. And I hate to say it, but I think the answer is that it's just too large an industry, and economically, too global. The areas where you need to slow down globally are relatively small. You can cross an entire ocean basin, go in 30 knots and never encounter a whale, and then the last 10 miles could be really important. And I think focusing on those areas is the most important thing to do. And if you're thinking about sort of gives and takes and trade-offs, not telling vessels that they need to slow down in areas where it's not prudent and where we can say that there aren't going to be a lot of whales is as important to show that it's a two-way street in that case.
I was just wondering, I love marine mammals, and I was wondering.
We're going to get to a mic so the folks online can hear.
I was just wondering, do you see that the health and longevity of marine mammals along the California coast, is it improving or is it consistently deteriorating? And so what is your perspective on that?
So the last commercial whaling station in the United States was shut down in 1972, just up the coast here in California, in San Francisco Bay. So we were killing whales up until 50 years ago. So the last 50 years has been an opportunity for a lot of these populations to recover. And we've seen moderate to good recovery of most baleen whale species when we leave them alone. Gray whales are basically at their carrying capacity for the environment. They tend to have these little episodic die-offs because they're pushing the limit for what the habitats can hold. Humpback whales have progressively increased to a point where we think that they are effectively fully recovered in this part of the world. Blue whales are sort of at a level. Their populations have kind of leveled off where we are. Some of that may be because it takes them so long to mature and to reproduce that getting that population growth is much slower. So for some of the whales that we can measure it in, we've actually done a pretty good job of allowing them to come back in the numbers in a population sense that are good. But clearly there are impacts that are increasing because as the whale numbers increase, you're going to see higher mortality. There are some species that are clearly not recovering and that are almost remnant populations, and those are really difficult to see. The dolphins, I don't think we really have a good baseline for how many there are, except for the fact that in the eastern tropical Pacific, in the 60s, 50s, and 60s, literally millions of dolphins were killed as part of the tuna fishing industry, where the whole process was to find a group of dolphins, set a large net around the dolphins because the tuna hang out under them, pull in the whole net, take out the tuna, throw the dead dolphins away. And that was the impetus, showing that, visualizing that to the world, was the impetus for the Marine Mammal Protection Act. And since that law enacted, those dolphin species have recovered quite nicely, or the mortality of those animals has minimized quite significantly. So,
Thank you. I apologize. My son is messaging me.
Oh.
Because it's past 7.
That's okay. We've got a question right here in the third row. We've had one. Yeah, yeah. If you don't mind, please use the mic.
Thanks. Thanks, Ari. That was a great talk. My question is about your very last point on collaborative and integrative research. So I met Ari virtually 2 years ago with 49 other scientists, or maybe 39, from 5 continents, or 3. And we were supposed to have an in-person meeting about how to detect and avoid North Atlantic right whales, which are endangered. We didn't do that because of COVID and the government shutdown, so we were online. And I was on the convening team, and our challenge to ourselves was, how can we find some people that aren't the usual suspects to talk about this? One of them was Ari. He was not a usual suspect. So my question for you is, what's your experience with that cross-disciplinary, integrative kind of conversation, and what are your hopes for the future around more of those to address the kinds of issues that you're talking about?
yeah, it's an awesome question. First off, I'm honored that I was thought of as one of those kinds of folks. So when I was at Duke, I did my PhD at Duke. I was in the ecology program, and for a long time, ecology wasn't even thought of as a discipline because it was too interdisciplinary, and it fell under zoology or biology. And 2006, the year I graduated, was sort of the first year that ecology was even considered its own discipline academically at that university. And to me, that's sort of a step change in our thinking that science isn't just singular disciplines. And I think since then, every student that's been trained since then, and especially at UC Santa Cruz, where you have majors that are much more interdisciplinary, you're creating a workforce of people that are coming out that have that broad disciplinary, not just within the natural sciences, but from natural sciences to social sciences. And I think a workforce is being created, and I do not want to be ageist at all here, but I think the people that are going to be coming into the workforce have that as a mindset already. And it's not going to take a lot of effort to create those kinds of communities. At least that's my hope. So that would be the passive way to do that.
And I think UCSC was founded on that. That's part of its DNA.
Yes. Yeah.
So I was an environmental studies major because it was the first place to have that major.
Yep. Yeah. That's important.
1972. That's awesome. So
That's awesome.
your
Yeah.
hunch is right.
Yeah.
Are we going to jump to the back? We're going to take one question from online, and then we're going to come back over to you.
It's a lot of pressure. There's a lot of questions in here, so I don't want to pick the wrong one. But I think one that kind of stood out, and it has been kind of harped on a couple of times, is, everybody's interested in marine mammals. We love talking about marine mammals. It's interesting to come to a talk where you get to hear about whales and sea lions, and then you get here and you realize, like, oh no, they're all screwed. And then one of the questions was, what can one person do to contribute to this type of research, and how can people who aren't necessarily directly tied to the field help out, and what can they contribute? And then another one was, what type of work or volunteer opportunities are available for undergraduates or early career scientists?
Yeah. Love the question. One of the classes I teach here at UC Santa Cruz is called Life in the Sea, and it is a introductory ocean class for non-majors, and I love it. And it is the best class because I get 130 kids that are not scientists. And the first thing I tell them is, if you're interested in the ocean and conserving it, we need scientists, we need lawyers, we need writers, we need any kind of discipline, economists, artists. There are politicians, yes. I mean, I can't think of, maybe outside of a couple of disciplines, a way that you can't affect change through that community and through that lens. And I would almost turn the question around and be like, how can you not? Rachel Carson, fantastic scientist and writer. I mean, I can give examples of every discipline out there that has a connection to the ocean and conservation, and doing your part with what you do best, I think, is the most important thing to keep in mind. And the passion and the interest is really what's the most important. Of course, if you're super wealthy and you want to fund and endow, science and students, that's a fantastic way to
See
do
me
it, too.
later.
Right. In terms of volunteer opportunities, if you're talking about from an undergraduate at UC Santa Cruz, I've been amazed at how well the university is set up to create paid internships for undergraduates in the summer. There's the Camino program, there's the Access program, which is sort of like an NSF REU. There are ways that undergraduates can get paid to work in labs, and I try and push undergrads always towards those things to get value for your work and for your time. It's hard to be a volunteer and a student. If you're out of student sort of time, being a volunteer is slightly a little easier. The challenge that both Colleen and I have in labs is that you invest a lot in a person to get them trained to do something, and you want to be able to see that person through and mature and matriculate. And sometimes if a volunteer can only be there for a couple of months or for a week or two at a time, it's hard for us to be able to invest enough in that person to make it a worthwhile experience for us. And sometimes we have to be a little selfish in that case. And
Practical.
Or practical, that's a good way to put it, sorry, practical. But if someone is interested, there's a lot of citizen science opportunities. I have a Fulbright fellow from the Ukraine who just gave birth to a baby boy about 5 months ago, and she started doing walks along the coastal campus and realized that she's seeing marine mammals all the time. And so she set up a schedule for herself where twice a week she would go out and do a survey of all the marine mammals she saw at 3 or 4 different points along West Cliff. We took that to the Seymour Center and basically now have docents and volunteers that are going out and twice a week going out and doing these little volunteer marine mammal counts. And we're going to turn this into, we've now got a year of data, and we can tell you what you can see from the cliff top, at the Coastal Science Center. We're going to put up a little chalkboard on the overlook and a pair of binoculars. Anybody who's out there, write down what you see. We can start to tally up these things and have people contributing to our science in a way that they can enjoy as well. Yeah.
We still have a question? We get a mic, we got 2 in the back row, and then I think Ryan will jump over to one more online.
My question is, how are the chemicals, the DDT and the PCBs, how are they getting into the whales? Is it through krill, what they eat? And similarly for sea otters, whatever the bivalves or whatever they are eating. And are we also ingesting it?
To the first part of that question, yes, it's through bioaccumulation of the things that they eat. They aren't really absorbing water through any other mechanism in their body, so it's largely going to be through what they eat. In terms of what we ingest, I couldn't tell you. I mean, I know mercury is really high in tuna and things that we eat, like swordfish, and we get that. But I don't know. I mean, luckily, we don't eat these marine mammals, so we're not getting those loads. And I think a lot of the animals that we do eat are shorter lived and lower, the more you eat high up on the food chain, the more likely you are to ingest these kinds of things. But I'm not a super expert
It is
on
kind
that.
of surprising. I had that same thought. I don't know if everybody had a guess when Ari presented the pesticide slide of which species would be most impacted. This is ongoing work. It's not published yet, right? I thought it would be the sea otters. They're really inland. They're
Right.
in an estuary that drains to agricultural areas. I thought it would be them for sure. So when the humpbacks came up as having so many contaminants, that's what got me thinking about the age differences and the lifespan differences.
Yep.
It's super interesting to think about how that transfer occurs. So Ari, I suggest you slip one of your own tissue samples in with your next with your next lot of analysis. Ari eats a lot of seafood.
Yeah.
He spends a lot of time on boats. Eats a lot of
I'll let
fish.
an otter biopsy me. Yeah.
I can
appropriate.
let Ryan shoot you with one of those little bow and arrows.
I'm fine then. His
back
aim is
there.
not that great.
Yeah,
Yeah. That's
yeah,
right.
that's
true.
right. Ryan, do we have, I know we've got a really large group of people online. Oh, wait, sorry, we're going to do one over here, and then we'll jump maybe to a final question.
Thank you. This is kind of going back to the marine protected areas and the ship strikes and, getting corporations and people to respect those. But I know fishermen, some very stupid fishermen, will intentionally kill like marine mammals that they come across because they see them as competitors for their fish. And so do you know if like other corporations or organizations, and I guess especially in our current political climate, have kind of those same dispositions for like, I'm trying to figure out how to explain this, have like the same mindsets for whether or not they follow like those protected areas and how fast they go and stuff like that.
Yeah, I've, for all the working groups and engagement that we've had with these kinds of communities, I've really never run across somebody that actively wanted to hurt animals to protect their livelihood. I think most people that I've come across respect and want to protect and conserve things, just not at without diminishing, their livelihood. And yeah, I've really never seen it get to that next level where somebody was that aggressive that they thought that removing that animal would increase their, their take home.
And that may be because you study whales, but tell you 50% of sea lions that strand in Southern California have evidence of gunshot wounds, most of them recovered, 50%. There's a couple, 200,000 animals down there, so it's a pretty high rate. We're going to go to the last question back here with Ryan, and then we're going to turn it over to George.
Yeah, another bummer. But I think, again, it's a very similar question in the same vein. If we kind of zoom out, a lot of people were noticing kind of on these whale tracks that they're going all over the place, right? And all sorts of different countries. And I think the general gist of the majority of these questions are, do other countries care? Are they reacting in the same way? And is our country's reaction enough? And what types of legislation would need to be in place to protect these guys moving forward?
Yeah, it's a great question. I'll push this one to the North Atlantic right whales, where population of 300 to 400 animals, it's critically endangered. We literally know these whales are getting hit by ships and they are getting entangled in lobster gear, and they go to Canada and they go to the United States. And there's one country that's moved shipping lanes, and there's one country that's thought about doing these things in earnest, and there's one country that hasn't. And we have done in our country a fair amount of work to try and solve the problem through technology and not changing operations of vessels, but making detection of these animals acoustically a way that we then say, let's just back ships down. So it's come from a perspective of keep the process the same, but try and slow it down. Canada has taken the alternative of let's literally just move the shipping lanes if we know that there is a risk there and that whales are getting struck by ships, and try and separate in space and time the risk to those animals. The problem is it's impossible to disentangle which one is working better in this case. And even with two countries, one animal, and a very straightforward interaction that we know is occurring, it's really hard to be able to figure that out. And so for a whale that swims through 7 or 8 different countries, from Colombia, Ecuador, Peru, Chile, Argentina, the Antarctic, that's going to even be orders of magnitude more difficult to try and get countries to all buy in and do things the same way. So it's a real challenge, and I don't know how to answer it other than to say we haven't done it great yet.
And just on behalf of Ari and I, while we're all here, we want to thank George and Rafe Kraw for the amazing opportunity, not just at this lecture, but for all of these lectures, all of the good science. and just bringing people together for such a wonderful reason. Thank you so much. It's so generous of you to do that.
Thank you. I want to thank you for a wonderful conversation tonight and invite you all to come back on February 5th when Professor Eric Polkovitz will be talking on transforming Pacific salmon recovery. Good night, everyone.